This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 467, recorded on November 7th, 2017. It's election day here in the U.S. The Department of Microbiology at the Icahn School of Medicine at Mount Sinai wants you to know about their graduate and postdoctoral programs, composed of over 20 virology labs, all centralized in one building in the heart of New York City. This department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to our program by the December 1st deadline. Interested postdocs are encouraged to contact faculty of interest. For more information about the department, please visit bit.ly/micromssm. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, Vincent. How are you?
I think we're in winter, man. It's like 9 degrees this morning.
Well, that may be true for you. We have 74 Fahrenheit, 23 C, cloudy. It's going to clear up in the afternoon. That's the way it's been for the next few days. However, there appears to be a cold front coming through. The forecast high for tomorrow is 53 Fahrenheit, which is weird to think about. At any rate, here we are in our shorts. It's all good.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's actually not too bad here. It was a little chilly this morning, but it's 45 Fahrenheit now, 7 Celsius, and a few clouds, a very, very New England autumn kind of feel. I just got out this morning and mowed the leaves. So.
Mowed the leaves. There you go.
Yeah.
All right, we have a special episode today. We have 2 in-studio guests. One of them's been on TWiV before. The other has not. And they have an interesting relationship between the two of them, as you will see. Let me welcome back from the National Institutes of Health, Jon Yewdell.
Hey, Vincent. Great to be here.
The last one, you were here last time, weren't you?
Yeah, we did it here. Yeah.
You and I in studio.
Yeah,
right.
I had given the talk to the students and in the room just outside, and then Steve Goff was there, and then we did the TWiV together. It was great.
That was episode 208.
Yeah. Got my TWiV bump from that.
Wow, that was a long time ago. I can't load the page.
Yeah, I know, it's amazing.
So I don't know what year it was, but it must have been many years ago.
A couple of years ago.
Yeah. And also joining us from the Memorial Sloan Kettering Cancer Center, Teddy Yewdell.
Thanks for having me over, Vincent.
You related, you two?
Yeah. This is,
I ran into him in Columbia.
I am his progeny. Yeah.
Oh, boy. We're going to find out how you...
Vincent's going to go for the dirt today. So
yeah.
Well,
Family Feud.
It's easy. It's really easy. It's really easy.
TWiV 208 was November 25th of 2012. So that's 5 years ago.
November.
5 years ago, right.
Yeah, time flies, doesn't it?
My God, it does.
We'll give you another TWiV bump.
Yeah.
Now, Jon has just released the book, because you wrote parts of it previously, right?
No, I mean, what's online now is all new.
It's all new.
All new.
Because I thought there was one chapter where it says previously published.
No, none of it was published.
You had a couple of articles.
The cartoons were
okay.
Some of those were published in the Nature articles, but no, everything is new.
How is it downloading? Do you have any
I have no idea, because aside from being an internet idiot, I don't think Google may not even tell you how many times people access it. Kindle, I can tell you, I was ranked 22,038 or something like that, out of 22,039 downloads.
That's in the How to Succeed section. Virology subsection.
So I'm counting on the TWiV bump.
I'm sure you got a lot of downloads. We plugged it on Twitter.
Oh, thank you. Yeah.
It was my pick, and I wrote a blog post about it. I've plugged it on other podcasts. Wherever I go, I tell people to.
Thank you.
Yeah. I even sent it to my co-authors on our virology book. I said, you need to read this.
Great. Yeah.
So, and you can't beat the price.
Yeah, no, exactly right. And, I hope to go viral.
The Martian started out a free download on as a Kindle I don't know how they did that because you said you couldn't figure it out.
I could not figure out how to set it for less than 99 cents.
But it was really downloading
Yeah.
well, and someone said, you need to sell this.
Yeah.
And that's when it
Yeah,
really
so
went
I
crazy.
cannot make any money on it because I wrote it as an official
Yeah.
duty that I'm proud of, and
Right.
I want to give it away for free. And I view it, if it's any good, it's probably my most important contribution to science. So I'm really happy I did it. It's not easy. You sit there when you start, particularly, and like, oh God, what is it going to be? And then you get into it, and it took me, I don't know, probably a year
To write it.
yeah. And then lots of times, and I know it's filled with mistakes, but not from want of reading and rereading and rereading. And one of my college roommates, who's not a scientist, he's a really good tax lawyer, really smart guy though. He read it and corrected it like I was in 5th grade. So it came back with red all over it, and that really helped. That's Tom Daly, a really good guy. So yeah, so that was my only editing basically, and my family read it, but you guys didn't have much feedback.
Yeah, it was good. I liked it.
Yeah. So
Well, I want to talk about it. I've got some
Yeah.
parts
Okay.
that I've highlighted,
Yeah.
and we'll go through. Remind us, since you were here, we probably went through your history, and everybody's read the book, but just briefly remind us. Tuckahoe, New York?
yeah, so I grew up in Eastchester, New York. Went to Tuckahoe High School. Not a particularly distinguished high school, even though it's a very wealthy part of New York, north of New York City. And then somehow, because of the kindness of my guidance counselor, I think I got into Princeton. Thought I wanted to be a general practitioner, even though my parents were telling me I was not suited for medicine in any way whatsoever. Regardless, managed to graduate from Princeton. Got interested in research only because we were forced to do an undergraduate thesis, and just by the luck of the draw, I wound up with Arnie Levine. It was a great lab. I was mentored by a PhD student named Art Levinson, who would later become the president of Genentech, and he, I mean, it was just fantastic. Yeah, it was just really lucky, and we talk science all the time, so I really got into it. Still wound up going to medical school as a medical student. Very quickly
That
realized
was at Penn, I guess.
At Penn, yeah, I was at Penn, and then I realized I was a fish out of water. And I applied. Penn, in their wisdom, always kept a few slots in the MD PhD open for people who wanted to do it after they had gotten to medical school. So I applied to that, was accepted, and basically then I finished in 5.5 years and did as little as medicine as was possible, which was not much. Penn had this really mature attitude for students that your education was up to you. And if you didn't want to take any graduate courses, more or less, that was okay. I graduated with just 13 months of clinical courses. And basically, it was one of those deals with the dean where you promise not to practice medicine and they will give you an MD.
So you've never touched a patient?
Never, thank God, never touched a patient.
And if you're on an airplane and they call for a doctor, you just shut up.
Yeah, I shut up. I know a little bit.
He gives out advice. He still doles out advice.
I do a lot of advice.
Actually, I know several MDs who are practicing MDs who have said that if somebody calls for a doctor on an airplane, they're not saying a word.
Yeah, because of liability issues.
'Cause liability and, what state are you over, and just...
Wow, really?
Yeah.
So
and also, most MDs don't have a lot of first aid type training. That would, they've got some pretty much what you'd get in a first aid course. So
yeah, you're probably better off with a first responder.
Yeah,
in
you're better off with an EMT or a
exactly,
paramedic, really.
Exactly right. Yeah. So never me.
What did you work on at Penn?
So I worked with Walter Gerhardt, and it was just really lucky. I got thrown out of one lab because the guy was leaving, going to the West Coast, and landed with Walter, who had been his postdoc. His guy was Norman Kleiman, really great immunologist, but was going to Scripps. And Walter, when I entered the lab, had just made the first monoclonal antibodies to viruses. So just scientifically incredibly fortunate, kid in the candy shop. And every single experiment you did was a new experiment because it was the first time you had monoclonal antibodies.
I think you wrote that in the book. Actually.
Yeah, so that was my so that was my thesis, and I got interested in CD8 recognition. And actually, Arnie had started me that as an undergraduate. That's even though everything in my thesis was a complete artifact. The intellectual problem was stated. How does the immune system recognize a transformed cell? In this case, it was a virus transformed cell, a tumor cell. So basically, I've been working on that problem and also antigenic drift, because we're back studying that since 1974. Yeah, so you can keep the same interest your whole career.
Sure.
And the field, of course, gets better, and the science. The cool thing about science, as you know, and Rich knows, I mean, the problems are there, and then the science is so much more powerful.
Sure.
Just the problems get cooler.
I think you went to Europe for a postdoc.
Yeah,
right?
So I did a 1-year postdoc in Europe with a guy named David Lane, who, along with Arnie, is credited with co-discovering P53.
David Lane, by the way, was a postdoc in the laboratory where I was a postdoc.
Oh, wow. At Cold Spring Harbor.
No, this was at Imperial Cancer Research Fund.
Oh, you were there.
With Lionel
With
Crawford.
Lionel, yeah. So isn't it weird that Lionel didn't get any of the credit for P53 and David did?
Yeah, they were puzzling over these extra bands.
Yeah,
on
exactly
their,
right.
T-antigen immunoprecipitates.
That's exactly right. There was this band,
Amazing.
and one of the tricks was P53 co-migrated with immunoglobulin heavy chain, right? So that's why everybody missed it, right? Because they co-migrated. And back then, it was so. But eventually they found it, and it obviously became a reasonably important discovery.
Now, I think after that you went first to the Wistar.
Yeah, I went back to Wistar as an assistant professor. Always a mistake to go back where you trained because they treat you like a graduate student, as I was.
Right.
So I had an R01, the major research grant, and I had literally no space to my own.
Right, I remember that. Yeah.
I was in a corner of Walter's lab and begging and borrowing, and they were hiring new people and giving them these nice packages, and I had nothing. No equipment, nothing. But I had a good technician. I rolled the dice, hired my first technician. She was fantastic. She later became the first postdoc of my first postdoc, so she was really good, Amy Yellen. So 4 years there, and then just kind of semi-randomly wound up at NIH, where I thought I'd stay 2 or 3 years, and then wound up 30 years later, same job.
Still there.
Still there.
Right.
Yeah, I love it.
All
So,
right.
yeah.
Now, Teddy, where were you born, and which of these travels?
I was born in Philadelphia, in Pennsylvania.
Oh, okay.
Yeah, 1985.
What's your earliest recollection of science?
Oh, that's a good question. I remember I told one of my friends, he asked what my dad did, and I was probably 5 or 6 years old, and I just told him that he worked with rats. And my friend was a really precocious young kid. He's super smart, and his dad was a lawyer, and so he said, "Yeah, my dad kind of works with rats too." That's probably the earliest.
That's
I think.
great.
Yeah.
Can we have a rim shot? Alan, he's competing with you.
Yes, that's good.
Yeah.
That's really good. And I mean, your dad is highly critical of the biomedical research establishment, and so I'm curious, at what point did you get really interested in science? You didn't want to be a doctor first, did you?
I started as a
Pre-med?
pre-med, exactly.
Oh.
Yeah. Just
Where'd you go to college?
Colgate University.
Oh, yeah.
Yeah, upstate New York.
Is that Hamilton, New York?
Yeah, exactly. Yeah,
Right.
Hamilton.
I always get confused, and then there's Hamilton College.
Exactly.
Is that in Colgate, New York? No.
Yeah. Oh, they're like 15 miles apart.
Yeah.
So, yeah. And then, I mean, probably like a lot of kids, I had a really good undergraduate professor, this guy, Ken Bellinger, and took a couple classes with him and just really liked it, and then ended up working in his lab. And I got to do a couple summer programs at NIH. They have a really great summer study program.
They didn't work with your dad, right?
No.
He worked with someone way better.
Conflict of interest there.
No, he wound up with a guy named Neil Young. And man, when you saw how Neil Young mentored his summer students, you felt like a complete loser failure because this guy was great. He was great.
I know the feeling.
Yeah, he talked to us, there were 4 or 5 summer students once a week at least.
Huh.
And I also got lucky because I was with a postdoc, this guy Rodrigo Collado, who was just like, an all-star.
So by then you're at NIH, so it's
Oh, yeah.
easy for you to just pop over to...
Yeah, I could see him, every now and then.
Got it.
Yeah.
Okay.
But we were in different buildings, and Teddy did not use my name. I mean, I didn't help him, so it was all legit.
Nobody ever said, are you related to the guy?
Well, yeah.
Must have.
Yeah. No, I mean, Neil had known him just because,
Yeah.
crossed paths, but yeah.
And so after you were finished at Colgate, what'd you do?
So after Colgate, I did a year in South Korea in this Princeton in Asia fellowship, where you taught, in this position, I was teaching at a foreign school. I was teaching high school science.
Oh,
yeah.
neat.
So that was cool. It was in this, city, Gwangju, in South Korea.
And that was you taught in English, right?
Yeah, everything was taught in English. I taught, biology, chemistry, physics, or science. Yeah, for a year.
Mm-hmm.
And then I came back and was a technician at Rockefeller in Gunter Blobel's lab.
Cool.
Yeah.
Yep.
And again, I was really lucky. I got paired with a postdoc who is now a professor at Yale in the cell biology department, this guy, Patrick Lusk. And I was just there for a year, and then I went to graduate school at Stanford.
Who did you work with there?
I was in the biochemistry department. I work with a guy named Aaron Straight.
Mm-hmm.
He's a chromatin biologist. So that's really, where I got most of my training is chromatin biology, cell biology.
And now you're a postdoc at Sloan Kettering, right?
Yeah. So I kind of took I knew I wanted to do something different than, the biochemistry and the stuff I learned. And we didn't have the opportunity. We were forced to do an off-topic qualification exam our third year, which before I started, I was like, this is so stupid. It's a waste of time. I want to start doing my, thesis work. And then I did it. I started reading about germinal centers and B cell memory. And I was just, completely enamored by it. And if I hadn't done that, there's no way I would have made the leap because there's a significant learning curve at the beginning. But that pushed me over the edge. And
So over the years, did you talk to your dad about a career in science at all, or? Yeah, I remember when I told him I wanted to go, because I like doing research so much, right? I like doing experiments, that I wanted to go, do a PhD. He advised me not to. He said, "What are you, crazy? Like, don't do this. Don't do this." But I think he also knew that I really liked it a lot and that I wanted to do it. And then, so then once I started, it was a kind of a different story. Yeah,
but yeah.
One of the quotes from his book: "For those seeking the most arduous and competitive career with the lowest salary, academic biomedical research."
It's in there. It's up there with musician and artist and
Yeah.
dancer, right? That's what we've turned it into. We're all basically Billy Elliot. So Teddy needed to dance.
Do you want to have an academic career, or you're not sure yet?
That's the goal right now.
Okay.
But I've definitely, when I started my PhD to now, I'm a lot more realistic about, what might happen at the end. Right now, it's just like,
well, according to your dad, the odds today of a PhD student being a PI is less than 1 in 10.
Yeah.
Is that for all PhD or just biomed sci?
Biomed.
Yeah.
And so the official statistic, I think, from NIH website is 1 in 7 or something, but I don't think anyone really believes that.
Yeah, the reality, it's hard to pin it down because how far out do you go after the PhD? And there is a survey of earned doctorates that has been tracking this for a number of years, but even that, you say, okay, well, this person is not a PI 10 years out of their PhD, but they could still become one.
Yeah.
So at a certain point, you have to cut it off and say, okay, less than 1 in 10 is a very reasonable.
I think people
direction.
think it's probably closer to 1 in 20.
Something like that. Yeah.
So has that number really changed significantly over time?
Yes.
Yeah, Rich, don't you think so?
Well, I just don't know. I think about how many people, I've known who got PhDs along with me who became PIs, and it's not a big number.
The data show this, that actually back in the 60s and 70s, you had almost a 50% chance of being a PI within a reasonable time frame after finishing your PhD.
Yeah.
And today that plummeted, and because we produced so many PhDs and the funding did not go up geometrically as it couldn't, and so we have this system that I'm sure we'll be talking about more in the rest of this episode that is built to overpopulate the world with PhDs and has done exactly that. And so the number comes down dramatically, and it's certainly less than 1 in 5 by the time I was coming up, and it's certainly less than 1 in 10 now.
Yeah, what I tell kids and what I remember was that when I graduated, which about the same time, Vincent was a couple of years ahead of me. You were in 79, Vincent, your PhD?
Yeah.
Mine was 81. Was that if you went to a good graduate school and you were okay and you weren't completely crazy, which did narrow it down probably to 1 and 2, you basically got a chance to be a PI, right? And so the kids who were okay and they came out of a good program, basically everybody got a chance. And you didn't necessarily succeed. It was easier then to be a PI, but it was still hard. But you did get a chance, and that certainly has changed. And I mean, Teddy went in with his eyes open about that.
Yeah, this is something that I actually warn people about when I'm invited to give career talks, that if you're talking to somebody from a couple of generations ago who's your mentor, and they're going to be right about a lot of things, but bear in mind that the world they came up through was very different from the job market that you're looking at now.
Well, I ask this question of myself all the time. So if I were doing it over again, what would have happened? Of course, I don't have an answer for it.
It depends when you're doing it, right? Now or back then, right?
Yeah.
Right.
All right. Well, I think we'll talk about this some more. Lots of things we can do. I wanted to talk a little bit about Teddy's work. So, by the way, if you want to look up his work, it's William T. Yewdell, but we're calling him Teddy. And
so he's Bill and Ted.
Yeah, he's Bill and Ted. So
this is his great adventure.
That's correct.
That's very good.
So looking at your publications, where you are now, looks like you're interested in class switch recombination. Is that a reasonable?
Yeah. My advisor, Jayanta, he did his Jayanta Chaudhary. He did his postdoc with Fred Alt.
Okay.
So yeah, he's coming, and he came from a very biochemical background for his PhD. So yeah, he was looking at DNA repair and class switching and this enzyme that does both of those.
So you, just to let you know, Fred and I were postdocs together at the Baltimore lab.
I didn't know that.
And I used to see him every Friday when they had group meeting. He would bring David Baltimore his sandwich. He would stop by his office. What do you want? And he'd go get it. And then sometimes the meeting would be going on, and Fred would come up and put this huge sub right in front of David. And then Fred came here to Columbia,
right.
along with myself and Steve Goff, all from David's lab within a few years of each other. And then eventually he left and went to Harvard, which is where he is now, and done fabulously, trained lots of people. And Steve and I are here. Steve is actually next door. So just a story.
The nucleus. You guys with the nucleus.
Nucleus.
Good start.
It was fun to bring part of his lab here, right?
Yeah.
That's the way I view
it. So many great people have come out of David's lab.
Yeah, for sure.
Yeah.
So tell us, what is class switch recombination?
Yeah, so class switching is, the antibodies are composed of, the very broadly the region that recognizes an antigen, and then the other side is, the heavy chain, and, different heavy chains can do different things. And so when you're a B cell and you come out of the bone marrow, you express both IgD and an IgM by virtue of alternative splicing. But what you can do is when you get activated, depending on the cytokines you see, you can switch from IgD and IgM to a downstream heavy chain, whether it's an IgG1, IgG2A, B, so on and so forth. And the idea being that depending on, the environment that you're around and what you need to switch to mount an effective response, that will kind of point you in the right direction. So in order to do that, what's really cool is that you make these programmed, DNA double-strand breaks in the genome, right, in the IGH locus, which is kind of crazy when you think about it, because dividing B cell, you're putting something in the nucleus that is its job is to make mutations, and when it goes wrong badly, and you get B cell lymphomas. So either there's that angle of understanding the mechanisms of how these breaks are targeted, and then also how those mechanisms are co-opted, or what goes wrong when you do get lymphomas. So those are things that Fred was interested in, Jayanta's interested in, and we work on a lot in his lab.
So that was a calculated evolutionary risk.
Oh,
right?
100%, exactly. Yeah, for sure.
Wow. So what aspect of this do you work on?
So the enzyme that instigates these breaks, it's called AID, or activation-induced cytidine deaminase.
I remember when that was discovered, it was a big deal, right?
Yeah. Hanjo discovered it doing a subtractive hybridization experiment. Yeah, and then it was shown, in genetic models and mouse models that it was absolutely required for class switching and somatic hypermutation. So yeah, we study the mechanisms through which AID is targeted to the IGH locus. And then more recently, we started a collaboration with my dad's lab, where we've been looking at, more generally B cell responses to things like influenza and some germinal center biology. And we can take, these point mutants, or even an AID knockout is really interesting for a lot of these questions, and see how those behave in these models. And so we've been seeing some interesting phenotypes.
There's a fellow from the Alt Lab here at Utya Basu.
Oh yeah, he just gave a seminar. The one that my dad gave yesterday, he gave last week. Yeah. So I met him for the first time. He and Jayanta basically, got him kicked off in Fred's lab. Yeah. They overlapped, and they were co-authors on a paper together. And they had almost identical tracks. They both are from Kolkata in India. They went to the same college. They did their PhD in the same lab. They did their postdoc in the same lab. And now they're both professors in the same city. Yeah.
He just got tenured not too long ago. He's done great. He started working with AID here as well.
Yeah, he is great. Yeah, I loved meeting him. Yeah.
So in general, if you take a mouse that lacks AID, I presume it lives for a while, right? If you keep it clean.
Yeah, they're generally okay. Yeah, they might have some problems every now and then, but they're pretty much okay.
If you infect them with a virus, is it a worse infection?
The interesting thing, it probably depends on the virus for one, but one of the first things that Hanjo did after AID was discovered with the knockout mice is they infected them with flu. And the title of the paper is that, IgM is sufficient to protect a mouse from flu infection and challenge is the title, right? A very vague title.
Yeah.
Because the mice were okay, and at least in whatever dose they gave them, they lost weight at the same rate, and they survived at the same rate.
So they can still make IgM antibodies.
Yeah.
Which, of course, we make for a couple of weeks or months before IgG kicks in.
No, IgG kicks in earlier.
Earlier?
Yeah.
And
Day 10, Teddy, day 14. You can certainly start to detect it by day 14.
The HA specific, by day 14.
Yeah.
Because that's how we figure if an infection is new or not, right?
Exactly.
Whether they're making IgM or IgG.
Yeah.
So, say survive. Huh? And you said it depends on the virus. Are there other viruses that are?
Well, no, I just say that just because I think if we, from my studies with them, and we haven't quantified this, but I suspect that if you give them a higher dose, they will be worse off than a wild type. But there is some interesting things. So the germinal centers, these sites where the B cells would, undergo affinity maturation, and the AID knockout has elevated germinal centers. It's like a hyperplasia, one of their phenotypes. So what happens in the flu response is you actually have a higher proportion of cells that are recognizing HA. And we're interested in finding out, why this is. But we think that kind of can give them a little bit of a boost, right, where they would otherwise be.
Yeah, sure. So in a chronic viral infection where the virus is evolving, what effect would that have on AID null mice, you think?
Yeah, I think that would be a really great experiment to do.
Because humans that are infected with HIV, the virus mutates to evade antibody, then the humans make more, and it goes on over and over. So would they make new IgM all the time? Probably, I guess so, right? Yeah,
I mean, right. So in these mice, they are forming, germinal centers, but they're not competing with each other in the sense that, they're undergoing, mutation, right? So they're basically just germline competing with germline.
Are there any animals that only make IgMs, or do they all have class switch recombination?
That's a good question. I don't know, Dad, do you know?
I think they are different. So, different animals, excuse me, different vertebrates have different antibodies, right? Yeah. So we had a really cool paper on lamprey.
Yes, I remember.
Right? I sent it to you. I said you guys should discuss it. So lamprey the cool thing about lamprey is they make antibodies, and they were discovered in the 60s by functional activity. Much like antibodies were discovered in the late 1800s. We immunize a rabbit with diphtheria toxin, and we can transfer the serum, and it's neutralized, right? So there's something there, which they called an antibody. So lamprey were known to make antibodies in the 1960s. It was a work of a very famous immunologist named Robert Good. There was a young guy in that lab who's Max Cooper in the 60s, who's still at it in his 80s, extremely active. And Max's lab discovered 10 years ago or so that lamprey, they make antibody, but it is not immunoglobulin. It's something called the VLR. What's a variable? What is it? A variable? I forget what it is.
Yeah, I forget what it stands for.
But it's something else. And they have a completely different way of generating diversity. And the cool thing is, and so thanks, Rich, because this is what Megan's first paper in my lab was. It's a great paper. What Megan showed was that if you immunize the lamprey with influenza virus, they respond in a very similar way that mice do, that humans do, that chickens do. Chickens are birds. They make antibodies. We can talk about those because they're a bit different. And not only do they recognize the same major antigen, which is still the hemagglutinin, and it's not based on molar quantities, because the most abundant proteins in the virion are matrix protein, which is basically not immunogenic at all, nucleoprotein, which is a little bit immunogenic, but HA is at the top of the list, even though it's the third most abundant, and neuraminidase, which is not very abundant at all, is second on the list. And this is what mice do. This is what people do. This is what lamprey do. And not only that, the antibodies in the mammals are directed pretty much exclusively against the head, not against the stem, which is this region that everyone's so interested in. Peter's lab, Florian Kramer, that we can make vaccines there. Great idea. But it's hard to generate those antibodies in mammals, and lamprey as well. They do not detectably respond to the stem. And not only that, the residues that we identified in the head as changing the antigenicity for antibodies, for immunoglobulins, also change the antigenicity for the lamprey antibodies. So the title of Megan's paper is something like that immunogenicity is conserved. It's a property of proteins, basically, and not so much apparently to do with the structure of what is responding to the proteins, because the VLRs that Ian Wilson's lab has beautiful crystal structures. They look nothing like an antibody.
They're like these modular, right, where you can have the components stacked on each other.
Exactly right, Teddy. Yeah. You can have 3 to 7 leucine repeats, and so they're crazy. Yeah,
VLRs is variable lymphocyte receptor.
That's it. Yeah, and so this is to me this is a fantastic discovery. And the lamprey. So these are jawless vertebrates. There's 2 of them left. They diverged 500 million years ago. The other animal that is in that group are hagfish. So these 2 guys have something completely different. Max's lab knows that they have something like T cells. In the genome is nothing like an MHC, so no one would know what exactly they see. And they have another kind of receptor that the proto T cells express, and I'm not sure where they are now in defining that. But so it's crazy because they split off 500 million years ago, and basically, they converged on a common kind of solution to the problem of having invaders, but with completely different molecular mechanisms. It's so cool.
Yeah, it's neat. Yeah,
so neat. Yeah.
And by the way, these the hagfish and the lampreys are really weird in a lot of ways. I mean, these are like fish out of a horror movie. That the hagfish produces this incredible amount of slime when it's threatened.
Ghostbusters.
Yeah, and the lamprey has this horrific looking mouth that's just a ring of teeth.
Yes,
and it sucks onto the side of fish.
Yeah. Yes, yeah. It's a nightmare. So when you work with a lamprey, and we didn't do this ourselves, it was a really good collaboration with a group at Emory. They don't use the mature lamprey, which apparently are not. They don't respond apparently very well to antigens anyway, because they're semi-senescent, is my understanding. You work with the larval form. So for up to something like 15 years, these pencil-like creatures live in the bottoms of waterway, and they filter feed. So that's what you work with, and you get maybe 50 microliters of blood out of them for testing. And you can't, they're wild captured, all of them. So fishermen capture them out of the Great Lakes, I think, and then you buy them from them.
Right.
Yeah.
Cool.
Yeah.
The Columbia River is loaded with lamprey. Yeah. So if you go, we recently did a little tour of the Columbia River where we went and visited some of the fish ladders in one of the dams, and you're underwater looking through these windows for the fish, and the lamprey are all sucked up onto the windows, and it's really creepy.
If you ever see pictures of whales, they're always hanging off the sides, right? Or other big fish. No,
I think those are remoras.
Yeah.
Yes.
Those are remoras, not.
But those are sharks, right, Alan? Anyway.
Yeah, I don't think remoras are a type of shark, and the sucker, it's a very different relationship.
Yeah.
Okay.
Yeah, I don't think mammals necessarily suffer from them.
And I think lampreys are fresh.
But it's, just.
They are, yeah. Okay.
Pragmatically, it's a huge problem in the Great Lakes because they were introduced. And so it's not native, and they're wiping out fish populations, yeah.
So that's a great example of how you can study a weird organism
Absolutely.
and learn great things. And, and Senator Proxmire would say,
absolutely.
What are we studying?
Yeah, and Max could not be funded for years. And to me, this is a Nobel-worthy discovery, a whole different system of receptors. And, you have to be an idiot not to think you could do something, even aside from how interesting it is, that they're going to be useful. So for example, we would love to have antibodies against ribosomes. Very hard to make them because the ribosomal subunits are so highly conserved. Well, your best shot is to go into a lamprey then, because there's a lot of evolutionary distance, right? And so for so many things. And you have to have such a limited imagination not to see how something like this is so important. But unfortunately,
idiocy is no barrier to employment in politics, apparently. So
or science.
The problem is that sometimes you don't know where something is going, but you just have a hunch, and you're a good lab. You should be able to work on it, but that's not how it works these days.
Here's the thing.
Right?
Any truth that is really true that you pry from nature is going to be useful to humanity sometime.
Yeah.
Any truth.
It's a good statement.
Right?
Yeah,
it's just, you never know what it's going to be, and even something so obvious like Maxwell's laws of electromagnetism. It took 200 years to do anything useful with it, right?
Sure.
And not to mention monoclonal antibodies. It was obvious from the day they were published in Nature, these were going to revolutionize medicine. And it took 15 years to go from that paper to the first drug. And even then, people were saying, oh, you guys have oversold this. You basic scientists, you're always giving us this bill of goods. This is going to be great. And nothing happens. And today, of course, more than half of the new drugs are monoclonal antibodies.
Yeah, sure.
Right?
It takes time.
Even obvious things take time.
Yeah.
And other things take much longer. And we know this song played over and over again. And, the enemy is not the politicians that I've seen from trying to work on science policy issues in DC. The problem is much more the scientists. who
The enemy's
run things.
inside.
It is.
Yeah.
It absolutely. It's pogo. The enemy is us. Enemy is us. People who want immediate translation of discoveries, and so all the money goes to translational. People like Teddy are pushed to do translational research when they're young scientists, and this is a big mistake.
But I think these are actually reinforcing things. I mean, so a significant amount of the blame does rest with the politicians for leaning on biomedical research to produce something. And I think that then leads to this internal policing of well, everybody wants us to do translational.
Yeah, so I was listening to C-SPAN one day a couple of years ago, and on C-SPAN was Newt Gingrich, who, you wouldn't necessarily think he was going to say something terribly interesting or useful. What Newt said was his biggest mistake as Speaker of the House was that he didn't double the NSF research budget, because the most important discoveries for medicine are going to come from basic breakthroughs in physics and chemistry.
Yeah.
And that was the smartest thing I had ever heard anybody say about funding basic research. And it came not from the NIH director, who any of them I doubt would ever say that. It came from Newt Gingrich, who in this one area has probably the greatest friend that biomedical research ever had.
When you can get him talking not on the campaign trail and not on camera,
Right.
He's actually really, really smart.
You've met him, Alan?
I've interviewed him for a story
Yeah.
a few years ago.
Yeah, he
and
is.
He's really smart. He's really thoughtful about science, and he gets it.
Yeah.
The problem is that then as soon as he sees some political prize on the horizon, he'll set all that
Yeah,
aside.
that's right.
And undermine all of it.
Yeah.
And he's now defending the current administration
Yes.
and their supporters, and it's painful to see that, because here's somebody who knows better.
Yeah.
Who is just deliberately, cynically exploiting a bad situation.
Well, I think my understanding is that he, more than anybody, was responsible for the doubling of the NIH budget in the
he certainly spearheaded it.
Yeah, and this other guy's my hero, who's I think it's just basically a good guy, is the representative from Illinois, Porter, who I met.
Yeah.
I met once at a FASEB get together, and he was impressive. He was the head of the House subcommittee in charge of NIH, and my understanding is that when they limited the chairmanship to term limits, he resigned so that he can continue his work on biomedical research, and he became the head of a lobbying organization in D.C. to fund NIH. And this guy spoke, and he was just brilliant. And there are politicians out there who get it. And what I could see as well is that the people on the Hill, the staffers, were really concerned about NIH research. They were absolutely concerned about how we treat postdocs.
Right.
And the problem is not there. The problem is the people who lead science is to get them to ask for the politicians for the right things. That, to me, is the problem.
Not too long ago, Newt had an op-ed in the Times calling again for doubling of the NIH budget. And I think it was during the campaign last year, in fact. And,
no, he is. This is the one thing this guy really, really gets.
Wow.
Yeah.
So, Teddy, what is your timetable? How many more years of postdoc you think you have?
I've been in Jay's lab for about 2.5 years. Okay. So, there was a pretty big transition where, I'm still learning so much about the field. It's like a different language coming from, biochemistry to cellular immunology. So I would say, 3 years, maybe 3 more years would be the goal right now.
Yeah. Okay.
Yeah. Just have to take it year by year.
Yeah, sure. And then start looking for jobs. Well,
yeah, exactly.
Good luck with that.
Thank you.
Wow. So there's a commentary on then and now.
Yeah.
That after 2.5 years, you're looking at
3 more
years of a postdoc.
Yes.
When I was doing this, 2 to 3 years was a postdoc.
Yeah.
Total.
After 2.5, I started looking for a job.
Yeah.
Sure.
Yeah. I mean, it's like my dad and I were talking about this. I've done really a lot of experiments and we have interesting data, but just the amount of stuff that has to go in packaged into one paper, right? Ridiculous. To get it into a big paper. It's compared to some of the stuff my dad shows me from his first papers. They were like figure 1A and 1B, and that's
table.
Done.
My first paper, Nature letter, two tables.
Yeah. Now it's,
took me 6 months.
7 or 8 figures, and then double that in the supplementary. So
right.
Building the story that we're working on now. My dad was giving me the advice, start sketching out the figures now, and we can do that. But it's just like to make it, to submit it to a high profile journal. If you want to be competitive, it takes a long time.
But as Jon points out, we've done this to ourselves.
We have.
Right.
Yeah, we are guilty of this. It's scientists. It's not the editors. It's us, in this case, the reviewers, demanding more and more. And I think, you enter this negative vortex where you send a paper in and people are not necessarily fair. And it just, you up the ante when you review a paper and it goes round and round. And we just descend into this place where every paper is so much work. And part of it is also the technology is much more powerful, ironically. So you do one experiment, you get more data than the rest of humanity ever got in the first 100 years of the field, right?
Sure.
I mean, literally. And I don't know, it's just crazy. And the costs are you wind up with a slightly better paper. That was a huge extra effort to put into it, right? Yeah. But the point rarely changes, right?
You made a point in your book. You talk about a paper that you had at Science for 18 months.
Yeah.
Satisfied the reviewers, but the editor was uncomfortable.
Yeah, exactly.
Yes.
You just want to choke them.
Now,
If that makes you uncomfortable, how about this?
Why do they have to be comfortable?
Exactly.
Shouldn't they listen to the reviewers?
They should. Yeah, that's another problem is that we allow professional editors who are well-meaning, dedicated scientists, we let them decide what's interesting and important.
Yeah, John, you continue to publish in, quote, luxury journals.
Yeah,
so why is that? You're established fellow. Is it some reason that you do that? Or
Well, yeah, so obviously, I mean, I have an ego, so I'm not going to deny that. So it's nice to have the papers there, but if you don't do that for your fellows, for your postdocs, they're not going to get jobs.
Right.
I won't say that's categorical. We've had people who have gotten jobs with solid publications because the committees were good and they actually read and interviewed the people and say, well, he's a bright guy. But opening doors generally requires a high-profile journals, and particularly if you're going back to Europe. Right. I mean, it's like there's a point system there, right? That they look at. And if you don't qualify, you're not going to get grants and therefore you're not going to get
hired.
Or
China. They need a cell paper.
Exactly. So
to get a job in China.
Yeah. And, we really harm science for this because, there's a big push now on reproducibility. And obviously all of us think that the work has got to be reproducible. But the problem is not that we don't do enough experiments or the p-values aren't good enough or we don't know enough statistics. And so we have like a nanny state now where when we submit a paper, we have to show all of our data and it's got to be open for everybody, which is crazy. I can't even go back once the postdocs have left and make anything of their data. So I don't know how a third party accessing the data online is going to make any sense of flow plots or whatever. I think this is just absolutely crazy. A huge amount of work for no gain.
I disagree with that completely.
Well, okay. The problem in science, according to me anyway, world according to GARP, is that it is too competitive. It's just way too competitive. And if you want to solve the problem with reproducibility, you have to start taking away the hyper-competitive nature of science, where every paper you publish, your career is on the line, right? Every grant you get or don't get. Mm-hmm. This makes bad people out of good people. You bring out the worst in humanity when you make it so difficult to have a career. And if I was NIH director, the thing I would worry about is reducing the competition to a more healthy level. I think competition is essential in science. That's how we test our ideas against each other. It motivates people to work harder, but you can have too much of a good thing, and we are way past that point.
Well, how do you accomplish that? That gets back to the 1 in 10. If you want to be a PI, you need to have these Nature papers. You need to keep on to that treadmill. And it's, I don't know, I see it as at least largely a product of the surplus of PhDs.
Absolutely. So how do we get back to that, Rich? How do we accomplish that, Rich? It's not easy, but I had a proposal that would accomplish it that was wildly unpopular, which was to make everybody basically an intramural NIH employee. Mm-hmm. Okay, so they're stationed at Columbia University, but like a doctor at Columbia could be part of the VA and be a federal employee. They're at a university. They still have teaching responsibilities. They can still have a lab, patient duties, whatever, but the government is actually paying them directly. And that controls the labor force and therefore the force that is getting money for research funding. So that is one solution to the problem, of which when I was trying to sell this 10 years ago to my colleagues, I think nobody liked that idea. But that is one way to do it, is limit the number of people who can be funded in some way. The problem we have now is that we can double the NIH budget, and then some dean at Columbia sees their career, opportunity, and holes appear in the ground. Vincent knows this. And then buildings show up. Yeah. And the plan is we're going to fund the building based on the overhead that we get from the grants from all the new people we're going to hire. And this goes on every cycle, this boom and bust. Every time there's more money, there's new buildings, there's new hires. And when we increase the budget, we generally make the problem worse and not better, right, for the competition. Yeah. So we need some sort of planning.
But regardless of how you're employing the PIs, there's also this problem that we built this system. I mean, Vannevar Bush came up with this system in World War II, which was built to rapidly ramp up research at universities. And before then, research was this kind of weird branch of the philosophy department, basically, that a few people would get PhDs, but it wasn't something that a lot of folks would go into. So this was all restructured in a system that is brilliant at generating a lot more scientists. And that is that your staffing and your training are the same process. So you staff your lab with people who are PhDs and postdocs, and you generate more PhDs and postdocs who can then go on to fill a dozen more labs. And that's great if you're in a period of exponential growth, as we were, and that won World War II, and so yay us. The problem is that after that, when your budget stagnates or at best doubles once every few political administrations, you've still got this process that is staffing the lab with PhD students who expect to go on and who have been, I realize a lot of folks deny this, but they have been implicitly promised a career in this field. And that's why they are willing to take a substantial pay cut compared to their peers who are going on to regular jobs in the workforce. And they find out that, oh, no, I'm actually not going to get that job unless I have a Nature paper, and so that feeds the hyper-competitiveness.
I do think that $35 billion a year, the extramural NIH budget, is still low for what we get. But I think a big problem is indirect costs. And, Trump tried to address this. He got vilified. But in fact, universities get anywhere from 50 to 100% of their dollar for every research dollar. And that's got to change.
Well, Vincent, at the same time, I was told by someone in Building 1, a policy guy, who had been on all sides of the scientific policy, for pharma, for universities, lobbying on the NIH side, and he told me the actual numbers are that for every 90 cents that a university raises in grant money, including the overhead, they actually have to go out and get another 10 cents. That they don't even cover the costs of the research infrastructure, even with the overhead costs. Now, bundled in there are all sorts of things like dean's salary and how big their staffs are. So the real numbers are hard to get at, but certainly they're not making a huge amount of money off of it, and maybe they're not even breaking even, right? And so it's always about the incentives. And so if you're going to be a successful dean, I think you're judged by how you've grown the program. And this is all part of the American mentality that, like, a company is never satisfied to have a certain market share. They have to keep growing to satisfy the stockholders. And this is all part of somehow the capitalistic system that we're in, right?
Well, I think part of the problem is here at Columbia, we have many faculty who get nothing in terms of grants, right? All the humanities, arts, etc., which are great programs, wonderful, but they bring no money in, yet their salaries are paid, their space is given to them. So we are in a way taking care of that, and that may be part of the reason why this 10 cents is needed.
Yeah, but this is just for research. I mean, those people, the kids pay tuition. They pay a lot of tuition here.
Yes, but science professors are part of what kids take. Yeah. So why isn't that tuition going to them? I think part of the problem is universities haven't tried to deal with this problem. They're getting a free ride from the NIH, and they really need to change. They will not, though, as long as they're getting money.
Well, then that's another way to break the system, because there is no other place to get money for the kind of things we're talking about.
No, not at all.
People will give money to put their name on a building or a lab. They will not give money to buy toilet paper for the infrastructure.
No, they will not. They will not.
The Jon Yewdell stall.
You wouldn't do that? How much?
I might.
I might do that.
Yeah, especially if it's the one you used, when you were a student.
Yeah, exactly. Old time's sake.
That's right. Teddy, you objected to someone your dad said.
Oh, yeah.
What was that?
That was just the, it is really annoying when you have to put, they say, if you put a figure and you have a Western blot and you're writing in the figure, legend, this is a, representative image. A lot of the journals, especially the big ones now, they make you put all three images in the supplement. And I think that's good, because myself, when I've been making figures, it made me go back and say, well, wait a second, let me go look at my other data.
It's a representative.
Exactly. And then when you're reading it, you can, as a reader, if you are really critical and you want to know what it looks like if you're trying to do the experiment, you get to look at 3 reps from them. And it does take more time, but for me, that's worth it.
Yeah. What do you think about this idea, Jon and Teddy, of limiting the number of grants that someone can have? That's not met with any kind of embracing, but...
I loved it. So I was actually in New York when they announced it. I was giving a seminar I was visiting Teddy. Teddy's got a son, my first grandson. So
cool.
Yeah, congratulations.
It's
Teddy.
Recent.
Yeah.
Yeah, he's 16 months.
Yeah.
So I come up
Thank
a
you.
lot now. And I was up here anyway giving a seminar, and they announced it. And I couldn't believe it, because to me, this was the first decent thing that NIH had done for young career scientists and for the better good of science. And I thought, wow, I can't believe this happened. And then sure enough, within 2 months, because there was this huge blowback, and what I understand, a lot of it came from the immunology community, which I'm kind of ashamed of because that's my main field. They backed down. So I was all for this, and I think the exact numbers weren't quite right, and the point system was kind of stupid. You get 5 points if you did a good service to be on a training grant. So, but all that could have been easily fixed, and maybe the number would have been 28 points or however. But the principle, I think, is really good. And so the old scientists say, "Oh, this is discrimination against us," right? Because these are the people who have all the money, and I think that's just ridiculous. The older you get in science, the more contacts you have, the more people owe you, the more damage you can do to them. And so it is a completely tilted deck in favor of older scientists, right? And so I thought this was a way of evening out the funding. And we all know, I mean, and there are published studies that the optimal lab size is something like 10 people, right? It's just on average, right? If we make a curve. And bang per buck, I think even the institutes know this. And there's debate about that, but it's basically that. And so we all know that the very big labs...
It's not going to be 50.
It's not 50.
Right. It's not.
The very big labs attract a lot of people, a lot of really good people, and those labs can run on the talents of 2 or 3 outstanding postdocs.
Yeah.
And we all know that, and you probably know that from your experience and Rich. And this was a way of addressing that problem and spreading out the money. And, there are also... There are all these issues that we've never really dealt with because it was all done by the seat of the pants. Is it a good national policy to have so much money spent in so few places, so few cities? Is that really a smart national policy? And yes, of course, the places like Harvard are going to be super productive because they soak up all of the talent in the country. And would it be a better policy, and would it be better for science? Would we have a better political grounding if the money was spread out more evenly over the country? And I'm not saying there's a simple answer here, because I understand that the problems are complicated, and I get both arguments, but these are things that have never really been debated, right? And I thought this policy was really heading in the right direction, and I was really disappointed when they backed down so quickly.
It was disheartening.
Yeah, this is a microcosm of a problem in the world generally, where you have a large amount of money flowing to a small number of people, and that small number of people, because of the large amount of money, wields an enormous amount of power.
Exactly right, Alan.
And because they wield an enormous amount of power, they are not going to let this system change because it's working great for them.
Exactly.
So when the older scientists say this is discrimination against us, what they really are saying is it ends discrimination in our favor. So the appearance of that, I mean, yeah, it could look like you're being discriminated against if you have historically enjoyed a tremendous amount of privilege and now that privilege is being leveled. But yeah, this would have been great. But as soon as it was announced, I looked at it and I said, that's never going to happen. There is no way that the people who have 50 people in their lab are going to let this happen, and they have the authority to do that.
Other industries have figured this out, right? Like the corporate law firms in New York City, all of them have, this whatever you want to call it, say it a senior staff problem. They have mandatory retirement ages because they have recognized as an entity, it's better for them as a whole if they make room for the young people to come up, right?
Yeah. And Teddy, what's the retirement age? 60?
It's 60, but they give you an option. A lot of them, I think, to retire at 60, or you can continue to work till 65. And a lot of them, they'll even continue giving you your salary until 65 if you decide to retire at 60. So They understand.
Just to get you out of the way.
Yeah, exactly. Yeah. But, these are capitalistic enterprises. They are solely there to make money, so they're going to know what the best way to make the most money is, and they've recognized this. So,
yeah, and there's also a well-established principle in management called scope of authority, which, from a number of studies, it's clear that nobody can manage a group of more than about a dozen people effectively. So they're going to have to delegate authority at that point. And that's kind of a rule of thumb, is if you're supervising more than a dozen or so people directly, you really need to break things out so that they're now supervising some of those folks. And so you get a lab that's bigger than that, and what are you going to do? And you're going to have to build some kind of a hierarchy. So that's nothing about this is an effective system, except that it has entrenched these inequities, and the people with the power to change it are the ones who are least motivated to do so.
So is there anything ongoing, Jon, that you know of that's going to address this issue, or is it dead?
Well, just what you guys could read as well. I have no inside information on this.
Right.
But what I read was that they still plan on setting aside more money for young investigators.
Yeah, right.
So I think that is good, at least.
That's great.
Yeah.
Yeah.
I mean, and, you know, the problem is that you and I had our own labs. How old are you, Vincent? 28?
29.
Yeah. So I was 29 when I started. And on average, investigators today are 38. And they get their first grants at 42. And if you're an MD PhD at 45. And I think I was at my best, you know, running a lab, although it was a tiny lab, but being at the bench with the people in the lab when I was 30. And I think we're robbing Teddy's generation of these years of running their own groups while they're still active experimentally. And in fact, it's so difficult now to run a lab that most people get the PI position and immediately they go to their office because they have to spend all this time raising money. And so the thing that they're really good at, which is why we chose them in the first place, which was doing experiments and thinking. And I don't know about you, Vincent, but I always thought better about the work when I was doing it myself.
For sure.
Yeah. And so we are robbing that of these people. And we're taking our most productive people and putting them in essentially non-productive labor, which is to raise money to do the thing we actually want them to do. And I think this is just completely stupid.
Yeah, I was in the lab for many years doing experiments, right? Yeah. And it was only much later that I stopped, but yeah, I loved it.
Yeah.
And that's why we got the job, as you said.
Yeah. And I, you know, I know, and I still go back and do some work. I think better when I can get my own hands on it, even though now I'm completely incompetent.
But
I was doing experiments up till the time I retired.
Wow. And Rich, didn't you think better when you were doing them yourself?
Oh yeah, I mean it's what I really loved. As a matter of fact, I have this. As I think about it, I think about all of the hassles and administrative crap going through my head and everything else, and I would go into the laboratory and sit down at the hood and just start splitting cells, and this calm would come over me, and I would go, yeah, and I would say, whoa, this is where I'm meant to be.
Yeah.
This is what motivates me. This is what I wanted to do. It's interesting because MDs practice by and large medicine throughout their careers,
right?
Because you have to,
right.
You need that. You need that experience.
That's right.
Doing the procedures. It's too bad we can't somehow, bake that same sort of attitude into basic science.
Well, will be another plus of shrinking the labs is the PI would have more time to work themselves.
Yeah.
My first 5 years, my office was a desk in the lab.
Yeah.
Yeah. Still, PhD programs still recruit 5-6 students per department a year.
Yeah.
Our department, one of many here, you had 5 new students a year.
Yeah.
What are they thinking?
Well,
I don't get it.
It's just what Alan said. We've confused our training pool with our labor pool.
Yeah.
Right? And that's the problem. And
you need those students to do the experiments to get your next grant.
That's the problem.
You don't have another mechanism to, I mean, you can have postdocs, but that's the same problem.
Yes.
You can hire technicians, which are, there's a cost associated with that, and it's not really viewed as a long-term career path by a lot of people, and it's not structured as such. So you need, I know this has been talked about a lot, even here on TWiV, but you need some kind of a staff scientist position that is a legitimate career path for a smart person who's ambitious and wants to go places, but doesn't necessarily want to be the PI. You need some kind of staffing like that, where you're not making more PIs, but you've got people working in your lab, and the system is just not set up for that, because you need to have underpaid labor in order to make the grants go as far as you need them to go in order to get the papers. And so we've got this chronic sweatshop attitude.
I mean, what was interesting, Dad, when he was talking to the students at Cornell and Sloan Kettering, he asked how many of you guys want to do postdocs, and there were 9 in the room. Only one of them put their hand up. And my old PhD advisor said the same thing at Stanford. Almost all the students who go in, my year, which was 7 years ago, we all wanted to be, PIs. Now he says the same thing. The vast majority of them don't want to do a postdoc. So I think it will be interesting to see if there is, if the system will self-correct itself. It'll take a long time to know if that will happen, but I don't know.
What are they going to do, the other 8, do you know?
They were just more open, whether it's industry or consulting and stuff like that, just other careers.
I just think this will never self-correct because the system, while is full of problems, as we've discussed, still produces amazing research. And, the deans and the chairs see this and they're happy to continue life as usual. Yeah. And Congress is, we've been flat on the budget for so many years now. It still works, even though there's a lot of pain and we've screwed up the publishing industry as a corollary. Yeah. It's never going to self-correct. I think we have to do something, but no single person has enough strength to be able to do it. As you saw, you made a suggestion and immediately it's shot down. Yeah. So I have very little hope that it'll ever change.
And in terms
I think the right NIH director could change it.
The NIH director.
The right NIH director could, because the one thing you have as NIH director is a bully pulpit. You don't have a lot of financial resources actually, but you have the bully pulpit. And if you have an inspirational leader like a Harold Varmus, who everybody respects, they can change the system. And Harold had a lot of positive changes, I thought, when he was director.
Yeah, but Harold's gone.
Yeah, so we just need another one. And I think one of the keys there is to get a really good director, an honest agent. They can't have spent their whole career angling to be NIH director. Yeah. What happened with Harold is that basically a helicopter came and showed up at the UCSF.
Plucked him out of the lab.
Plucked him out of his professor chair and dropped him in the chair at NIH. Yeah. Because he was a Nobel laureate. I think Clinton wanted to have a Nobel laureate he could chat with. And then Harold, he didn't have any political experience and he was a pure scientist, as we all know, a great retrovirologist. And man, he showed he had all these other skills. And I think there are people out there like that, well-meaning people who are science and love science and they want to do something for it.
Yeah, well, it's not happening in the next 4 years, I can tell you that.
No, we have to be patient, right? And it's going to take a transformational, I think, director to do this. And so maybe it's a once in a lifetime, once in a career, for all of us. It's probably too late for us now, but hopefully in Teddy's career, someone like that will come along.
Yeah, well, I'm not concerned about us. I'm concerned about the future of. It's all about the science. I learned a long time ago. It's not about us.
No, it's
science
not.
is what's important.
Absolutely.
And I don't think it's healthy at the moment, and it could be a lot better.
Agreed.
Rich, you and I, when we talked to Tony Fauci years ago, we asked him what. R01s are going down in number, and I think that's the best grant mechanism.
Me too.
Not these big centers with tons of people. Yeah. And we said, "Aren't you worried?" And he said, "We keep our eye on it. We're not there yet." So he wasn't even worried. Yeah. Yet I think now it's horrible, and he's still,
Yeah, I agree. I think Tony's been he's my director. I think he's done an incredible public servant, but this is something I don't agree with him on. Certain viruses, they put a lot of money into this kind of centers of excellence. They have really good people, many of them, but I think young scientists really suffer when the money is distributed this way because you're in a center, you have everybody else's opinion to deal with. And basically, if you're a young scientist, you make a great discovery, often it's going to threaten the discoveries, if not the careers of other people in your field. Yeah. And it is not a supportive atmosphere when your funding depends on having these guys along with you. It's not a good way to do it. Yeah.
Teddy, do you think that these issues are more real to you because your dad talks about them all the time, or you think you would have picked it up anyway being in the field?
Yeah, I think, it's something you would just get through osmosis because you see your advisors like writing grants, right? Sometimes you see them stressed out. You can tell, right, when the deadline's coming up. And so you will always talk about these things. But it definitely gets amplified because these are conversations I have with my dad a lot, trying to come up with creative ideas. But then really it just comes down to, you could have the best idea, like we've been talking about. What is the motivation behind that? And are the people in power willing to do that? And it's always no.
Yeah, remember that you guys had a TWiV on it when that what I thought was a great report from the PNAS came out, and Harold was on it, and and I thought, okay, man, these guys are behind it, and now maybe Bruce Alberts, and then it got such negative feedback from the powers that be. Yeah, right. It's just what Alan was saying. No, totally.
I mean, there and Arturo Casadevall regularly writes wonderful articles on what's wrong with publishing.
Yeah.
Just same thing. What's wrong with biomedical research funding?
Yeah.
And nothing ever happened.
He's one of my heroes. I think this guy has got the column he writes for the MBio. I think it's fantastic.
Yeah, for sure.
Yeah. So Arturo. So there's a guy, right? There's a guy who's got great leadership potential. And I think if someone like that,
We're NIH director.
even NIAID director,
Sure.
I mean, someone like that could change things. And so I don't think it's hopeless. I really don't. What I've seen, and we see this now, there's a saying in many countries, the fish rots from the head. Okay. And also means that if you have a good head, the fish prospers, right? The fish does well. And so the head of an organization really, really matters, right? In a way that I think many people don't get.
Yeah, I am not as optimistic as you are. I think we have a real problem here. And I think part of the issue is the general public does not realize what science does for their lives.
That's right.
And that's one thing we're trying to do with these podcasts.
Yes, absolutely.
But we don't reach enough people, right? And I do think it is the scientists who need to be saying, hey, this is what I do, and this is why it's good for you. And we don't have enough of that.
We don't.
So I do think if the general public were a little better educated with respect to science, they would put more pressure on their elected
I think that's actually
officials.
a separate problem.
Separate from what? From what?
I mean, the general public has an appreciation for what the military does for them.
They
and
do.
Yet we can't fix defense contractors.
Yeah,
right?
but they
And
get a
it's
lot of money, though. Military gets a lot of money.
Exactly. And it's exactly the same problem. The people with the power to change the system are the ones with the least motivation to do so.
Yeah.
And so we continue doing exactly what Dwight Eisenhower told us not to do and feeding this vast military-industrial complex, building up a military that's bigger than any other in the world in dollar terms, and that still can't win the conflicts that it needs to win because they're not operating in the traditional fashion that it's built for. And so I think there's an appreciation in the public for what the military does, also an appreciation that it gets a lot of money, and yet we can't, have nice infrastructure and good healthcare because we're spending the money on bombs. Science is a smaller version of the same story. I think there's a limitation to what the public understands about science, and that does lead to problems like the anti-vaccine nonsense that we talk about a lot. But it's separate from the structural problems within science, which I think can only be fixed from within science.
Yeah. So Alan, it's really a problem for game theorists, right? The mathematicians.
Yes,
because
exactly.
it's basically a game.
Right.
Capitalism, how we fund the general good. It's basically a game problem. How do we change the rules to come out for a solution that's more optimal for the most people, right?
We have a market failure here. Yes.
It is. And so maybe the mathematicians have to figure this out for us.
Maybe.
Because it really is a game.
Yeah, imagine if you took the NIH budget, right? This is just a dream scenario, and you multiplied it by 10 for a decade, right? Imagine all the discoveries that would come from that, right? You would think it would be crazy, right? Everyone gets a grant. You can do whatever you want. Yeah. Go.
I think it would be fabulous. But, your dad
and Teddy,
would say,
it's a time machine. We're 1970, because basically that's the way it was.
Your dad would say,
then 20 years from now, we'd be exactly in the same position.
No, because universities would build buildings. That's
exactly right.
Point,
yeah.
and expand.
Yeah.
So we have to fix that first.
Right.
That's right.
Yeah, any sort of significant expansion of the NIH budget has to come with some regulation, recognizing these problems that happened in the past.
That's right, Rich.
And put the money into RO1s and that kind of stuff rather than buildings.
I mean, we do
PhD population control.
Yeah, I agree.
Yeah.
We do need a visionary NIH director. I think.
do.
But he also he or she has to have a reasonable administration. I think you remember that at the outset of this administration, it was suggested to cut the NIH budget by $17 billion.
Yes.
This is just pure stupidity, as you would say. Right?
But it is, Vincent. But that number, my understanding is that they just looked at what overhead was, and without having any understanding about what actually funds the system, just said, we're just going to cut that.
The overhead,
so there was some idiot who just looked at a budget, had no understanding of where the money went or how dependent the university system is on it. Yeah. Right. Although what it looks like now is that the universities are being targeted. By the politicians, the influential universities with this new tax bill. So things could get really ugly here, right?
Yeah, for sure.
People need to vote.
Today's election day.
Yeah, people need to vote.
Vote. John, what have you done lately that you're really excited about?
Two things. So One got back into flu antigenic drift in a major way, and this is how Teddy and I are collaborating. I realized that, I had worked on immunodominance and CD8 T cell responses for a long time, and there's still a lot to discover there, but it dawned on me that we know virtually nothing about immunodominance in B cell responses, antibody responses. So what immunodominance is this tendency of the immune system to respond to various possible antigens in a hierarchical manner. So T cells, if I know your HLA type, I can predict pretty accurately if you get infected with flu, which exact peptides you will respond to, right? And can make a prediction. That's going to be reasonably accurate. In inbred mice, perfectly accurate.
The same goes for antibodies as well, right?
No. So antibodies, like nothing is really known about that, right? So I just thought, okay, this is something I can address. I'd like to do this before I retire. And so I had this idea that we could make a new kind of a panel of viruses. Basically what I did for my PhD, but times a lot. So what we do is we start with a monoclonal antibody and we select for an escape mutant, right? And then we do that again and again and again and again and again. And in the end, we have 5 major antigenic sites on the influenza HA, which is probably the most important part of the virus for a vaccine. And we mutate 4 of the sites, leaving 1 site. And we do that 5 times, right? So we have 5 different viruses now we call delta viruses because we've delta changed 4 different antigenic sites, leaving 1 site. And just experimentally, this makes it really easy to measure antibody responses to a single antigenic site. And now for the first time, we can study immunodominance at the antibody level, right? So antibodies, ironically, are harder to study than T cells. T cells took 70 years to figure out how they work. But at the end of the day, a T cell sees a short peptide bound to an MHC molecule. And so that's really easy to count them because you just make the peptide and you have the right MHC. You can count them quite easily by flow cytometry, other methods quite nicely. Antibodies, ironically, despite being discovered first, they're much harder to study because they're in solution. And it's complicated, and there are thousands of them in solution, and they compete with each other for binding, and their epitopes, the things they stick to on the proteins, are much more complicated. They cannot generally be mimicked with peptides. You have to have a folded molecule to do that. So you have to work with an intact structure. And so we can do this genetically. So we made this panel. There's a fantastic postdoc in my lab, Davide Angeletti, who did this work. And we published a really nice paper stating, I think, for the first time what the rules of antibody immunodominance are at this level of antigenic sites. And we could show for the first time, sort of in inbred mice, what are these rules? And that actually is also how we're collaborating with Teddy and many other labs, because this panel is really valuable. So any B cell person who's interested in some aspect of B cell or antibody biology can take our panel that we're sharing, of course, which we should do. They're published reagents, and they can take this panel and they can now map out the specificities using quite robust assays, even something as simple as hemagglutination inhibition or ELISA assays. And so we really can put immunodominance on the molecular level. So we did that in a classic mouse strain, the PRH strain, in mice. We're in the process now of making an equivalent panel for currently circulating human viruses. And we'll be able to get clinical samples, and we'll be able to study human beings now for what the exact composition of their response is to the various parts of the hemagglutinin. So that's one of the things I'm excited about. The second is a long quest starting in the 1970s to figure out how these CD8 T cells recognize virus-infected cells. And something we've known for a long time is that most of the peptides are generated co-translationally. So you have a viral protein like nucleoprotein from flu. The half-life of this protein is not measurable in infected cells because they die before you can measure any degradation. The half-life is something like 2 days, which is average for a cellular protein. And yet, you start making nucleoprotein, you start making peptides immediately. So throw flu on cells. If you have good enough T cells, the T cells can recognize the infected cells within 30 minutes. It's amazing, right? Where the hell are the peptides coming from? Because they're not tracking with the degradation of the protein. So we had this idea 21 years ago, that the peptides are coming from something we called a DRIP, a defective ribosomal product. And the idea is that basically Murphy's Law as applied to genetic transfer of information. You start with an RNA, you have to make an mRNA, It has to be decoded by the ribosome. You have to have the right tRNA. It has to have the right amino acid. It's got to fold correctly. The ribosome can't screw up. It can't start in the wrong place, can't stop in the wrong place, can't frame shift. The idea is that all those things can go wrong and do go wrong, and that's where the peptides come from. Okay? So we've been tracking that down for a long time now, and not just us. There's another great lab on the West Coast run by a guy named Nilab Shastri, and what Nilab has shown is that there's all sorts of alternative translation that goes on. And Nilab shown that there are many peptides that can start with a CUG, which is not a normal start codon, right?
Right.
And known for viruses actually before Nilab's work, but what Nilab showed was that the CUG can actually be decoded as a leucine, and there's a special tRNA for that. Okay, so it looks like that alternative translation, the CUG and these other things that we've been studying, are really where a lot of the peptides come from, particularly from viruses. So we've got a really cool project now. Excellent postdoc on our lab, JJ Way. And JJ is working on an old idea of mine that may finally come to fruition, that there's a subset of ribosomes that we call immunoribosomes. It's kind of tongue-in-cheek because there's an immunoproteasome that are dedicated to making peptides for immunosurveillance. So we've got some really cool findings now where we can knock out individual ribosome subunits. The cells are fine. They make viral proteins at the same rate, but they either increase the generation of peptides or decrease.
Yeah, that's very cool.
Isn't that cool?
I love it.
Yeah. So, and we're working with viruses with this, but we also now have a great collaboration with a good friend of mine at NIH called Lou Stout, and trained with David as well, actually.
Yeah,
just after you left.
Yep.
Yeah. And so Lou and I are looking for, Lou is the world's expert on B-cell lymphomas. And JJ now has got this ribosome profiling technique, this amazing technique from John Weissman's lab. One of my favorite papers of all time, describing the deep sequencing of protected fragments of RNA by the ribosome. So you can tell exactly what the ribosome is translating. So if it's translating crazy stuff, we'll find it. So we're looking at lymphoma cells. Yeah. And we're looking for new targets now because cancer immunotherapy is something that has arrived and is curing people now, right? And so this is the beauty of basic science, right? So most of the antigen processing work has been done in viruses, a lot with flu, actually.
Yeah.
And it turns out, of course, that cells being cells and bodies being bodies, what's true for viruses is also largely true for cancer.
Now, what about animals without antibodies and cellular immunity, like insects?
Yes.
Their ribosomes shouldn't do this, right?
Yeah. So who knows? Very good point. Their ribosomes should not do that. Yeah. And that's really hard to test. It's actually, if you try, we had an old paper on this, 25 years ago, where we expressed, class 1 molecules in insect cells, asking that very question. And they do not like to assemble class 1 molecules. So there are specialized, and we thought we put in everything we knew about them. We don't know much more now. Something missing about, it's not just simple erector set here where we could put the components together and they make it.
Right.
So it's a very good experiment.
That's cool.
That's right.
Somebody will do it because I like this idea that ribosomes have lots of functions that you wouldn't even think of. Like this drip is great.
Yeah.
Well,
Sean Whaling, right? Sean had this great paper showing you can knock out one of the ribosome subunits. They screen them all. And you can selectively affect VSV translation and not touch most of the cellular messages. I think Sean had the first paper showing that. And there's a growing field now. It's a woman at Stanford named Maria Barna who's fantastic. A growing field showing that, listen, a cell has about 3 million ribosomes, something like that, an average cell, right? And the ribosomes are at the heart of everything. They're the single most energy intensive aspect of what cells do. Most of the energy goes into protein synthesis. It's incredibly energy intensive. There is no way you have something so evolutionarily conserved that you haven't come up with a zillion tricks with, right? It's got 80 protein subunits. There's no way you don't play around with that. And you don't have some ribosomes missing some, others where you have, it's known that like S6, a very famous one in viruses, you phosphorylate it to control translation. We're just scratching the surface now of ribosome biology. And with viruses, there is so much, of course, a lot of the basic breakthroughs in translation research came from virology.
Absolutely.
Right? And your textbook on that, your chapter, translation is fantastic.
We, here on TWiV, we are firm believers in viruses illuminating the way.
Me too.
Right?
Yes.
Yeah.
Teddy, you read any good books lately?
Oh man, not since Finn was born.
Really?
The last really good book I read.
Have you gotten any good sleep lately?
Yeah. Sleep actually is not a problem.
Teddy, you were
now.
just this morning. You're just reading like Big Cat, Brown Cat.
Oh yeah, that's
like
true.
That.
Yeah. Brown bear. Yeah. What do you see? There was this great book. Dad, you read it. It was the one by the guy who wrote the
Finnegan.
thing. Yeah, by this guy William Finnegan.
That was great.
It was called Barbarian Days.
Yeah.
It was basically like an autobiography. He had originally written a piece for the New York Times. He's
The
one
New
of
Yorker.
or the New Yorker. Sorry, yeah, he's one of their staff writers. He writes politics, but he also writes about every now and then surfing, and he just has like a wonderful prose style, just like simple but just hard hitting. And he talked about his life in surfing, and that was like one of my favorite books of all time.
Teddy's a surfer.
Is that right?
And so he
Amateur.
But I'm not. I've tried once and fell off and hit my head and never did it again. But the book is so well written. I loved it. And what's funny is that there's actually really good surfing on Long Island.
Mm-hmm.
So some of the book is in the Rockaways or out on Montauk.
Yeah, the end when he
and New Jersey
married
actually.
and had a kid. Yeah, he talks about transitioning to a more urban surf life.
Yeah.
Kind of modest waves, though, compared to
Not.
elsewhere.
It's all about the storms.
Yeah, that's true. When we have hurricanes, they're out
Absolutely.
there.
Or nor'easters, right?
Yeah.
Teddy's been to Rockaway when it's been good.
Yeah, it can look like, rarely, but it can look like the Bonsai Pipeline if the conditions are right.
So that's crazy. You could take a subway to go surfing.
Yeah. No, that's good. I've seen people on the subway with surfboards.
Exactly.
I always wondered what they were doing.
Rockaway.
So there you
Follow
go.
Rockaway.
There's subway surfing.
In fact, I took a
Oh, yeah.
yeah, right. John, if you hadn't been a scientist, what would you have done?
Probably a businessman or something.
Yeah.
I don't know. Yeah.
Really, you like business?
No, I don't know. I'm Jewish. My father was a businessman. I sort of probably have it in the blood. I don't know what I would have done. I guess medicine, but I would not have been happy. I just didn't like it.
Nobody in medicine is happy these days.
Not too many, but something that may help young readers. I have a medical story to relate.
Mm-hmm.
It should be good for young people. So I had a family history of heart disease. My father died at 55 and his father at 55. And I'm not a very good athlete. Teddy's an exceptional athlete. I'm not, but I've always loved sports and was always doing endurance things. Like I've ran marathons. I do a lot of biking. And 3 years ago, I just out of the blue, basically, I noticed when I was exercising that, I was short of breath. Right. And I just had flu and thought, that's nothing. And then the short of breath got worse and worse, and I could go out running, and I'd run downhill, but I couldn't run uphill. Okay, so being an MD, in name anyway, I went to textbooks, and I figured out, okay, I have pericarditis. So I had very mild chest pain. I could treat it with ibuprofen. I had the shortness of breath. Okay, I'm just treatment. You just wait for it to go away. Okay, so it got worse, and my wife was an MD, Teddy's mother, pediatrician, and she was urging me, not that urgently, but go see someone. But you're an MD and you think it's not a problem. So one day I was riding into work at the end of it and just out of breath. I ride bike to work every day. So go to the ER, go see a cardiologist the next day, do that. Family history, I think you just need an angiogram. So I got an angiogram 4:30 in the afternoon and 5:30 next morning, I'm getting wheeled in for a quadruple bypass. Okay. So message to young people, particularly young men, if you've got a family history of heart disease, pay attention to it.
Yeah.
And I should have been probably checked out every 5 years. Teddy will be, his brothers, even my daughter, just get checked out by a cardiologist, because if I had been on a statin from age 35 instead of age 62 after a surgery, I probably could have avoided this. Right. So just you guys out there. And something else was the surgery. I had a great surgeon. I was home in 3 days. I was actually back on the track jogging very slowly in 2 weeks.
Wow.
And a key to that was that I was so baseline healthy because of my lifelong exercise habits, right? So just you guys out there, you young people, it's in the book, something I give in the talk.
Sure. Yeah.
Try to get exercise every day.
Yeah, I read that.
Every day,
I haven't started
You'll be happier. You'll be happier because mentally it's good for you. But when something comes along later on, and listen, no one lives forever. You're going to get sick eventually. And maybe it's 60 and maybe it's 70 or 80 or maybe it's 40. You're going to deal with it much better if you're healthy.
When I go on vacation, I walk a lot and I always feel better.
Yes.
For a week, walking just for a week, once a day, a couple miles. And I wish I could get back to it when I come home, but I never do.
Everyone should do it. I mean, I think it's really the key to a healthy and happy life. And fortunately, that got transmitted to my kids. They're all, to various extents, they're all into exercise. So,
and now you're okay, right?
Yeah. Now, that's a great thing about surgery. Particularly that operation. They are so good at that operation now. So my odds of making it through that, I looked it up afterwards, were 999 out of 1,000. That's how good it is. Wow. Even though your heart is open and you're a heart-lung machine, but they really have it nailed now. And the recovery was great. I mean, the nurses were just fantastic. And boy, you go through something like that and you love doctors.
Yeah. Surgeons, anyway.
When was that, John?
Absolutely. So I had it done at Suburban Hospital, which is
when?
2.5 years ago.
Yeah.
2.5 years ago. And, this guy saved my life. And so that was a very, very positive interaction with the medical system. And it's a thing in the U.S. I mean, if you've got good health insurance, the system here is unparalleled.
It's the same when you have a kid. When these experiences, basically, like when you go into a hospital and you assume you get good healthcare, but when it's actually you or someone you know, and you see all this crazy stuff they're doing, you're like, they're opening his chest, and you're like, oh my god, they make sure it doesn't get infected, and then he's back in 2 weeks and walking. It's just really amazing. Yeah,
well, he's also young. It helps, right?
Yeah, it does. But it's impressive when you're right in front of you.
Yeah. Teddy, do you have any scientists that you admire in particular?
Of course, my dad. He's my number one role model.
I did not pay him.
Yeah, not pay me. Yeah,
there's no threat of the inheritance.
Yeah. My PhD advisor, is also really high because I think you just have a special relationship with them because that's where you really get your chops from and you learn. And he really taught me how to just analyze data extremely thoroughly and especially not to overconclude, right? And just Aaron Strait be one of them, and
he is a great guy.
Yeah,
I met him and it was like, wow. And I thought two things: one, this is a great guy, and two, Teddy can really pick mentors.
Yeah, right.
Yeah, because he is fantastic, and I was super impressed by Aaron.
Yeah, and then getting into the B cell stuff, I think. Klaus Raesky, just his papers I really just love. They're just so creative and hard hitting, high impact, but also just like simple and elegant. So I love reading stuff from him. And there's a couple other labs, but him in particular.
Cool, that's neat.
Yeah.
Hey John, if you could have dinner with one person, living or dead, who would it be?
That's a good one.
My children.
Probably don't do it very often.
Right? No, I do. But two of them live in D.C. I mean, they're my favorite people. So if that's my honest answer, I have 4 children.
Yeah, 4.
Any of them.
What stages of life are they?
Teddy's second oldest. My oldest is a daughter, works for the Justice Department, so-called Justice Department.
She a lawyer?
She's a lawyer. And third one is working on immigration policy at basically a lobbying firm in DC, and the last one has a life of Riley. He lives 4 blocks from the beach in San Diego, and he works in a marketing firm. So he's got a really good life. But these are my favorite people, to be honest, and that's who I would choose.
That's a good answer.
Yeah, that's who I would choose.
We ask that a lot, but I don't think anyone ever said their own.
No,
kids.
It's a great thing when your kids grow up, and you know this, and Rich knows this for sure. I mean, if they turn out reasonably okay, and you haven't screwed them up. It's a great thing, and there is just nothing better than spending time with your adult children. It's my greatest pleasure in life.
Yeah, Rich does it. He's always traveling around the country.
Yeah, I understand it, Rich. I mean, it's fantastic. And then when the grandchildren come along, it's just the cherry on top, right?
Yep.
Yeah.
So he's still there. He's still there.
Yeah, I'm still here.
Yeah. So it's great. Yeah. So that's who I would choose.
Good answer.
Yeah.
No, you don't have to apologize.
Yeah.
You don't want to apologize.
Yeah.
Right?
Yeah, no, that's a good answer.
Yeah.
Rich or Alan, you have any other things you want to ask these two before we move on?
Well, I do.
Go ahead.
But it's probably a whole nother TWiV. John has this chart in his book that's productivity versus time spent on the job.
I love that.
You like the milli papers per week?
Yes.
I'm very proud of that, Rich.
I think you need to adjust your unit, though.
I think it's right.
No, it's like 50 papers a year when you're max.
No, it isn't. It's milli papers per week. Yeah. Yeah.
At any rate, I'm sure that this curve is different for different people in different circumstances, but it goes up and it, I fundamentally agree with the shape. It goes up and it peaks at 80 hours a week and then drops off because you can't do anything more than that.
Yeah. Well, it drops because the person is crazy after they're working 80 hours a week to levels of psychosis. But yeah.
I'm concerned about the perpetuation of this idea that in order to succeed in science, you have to sacrifice the rest of your life.
You don't.
Well, this is what I'd like to hear your comment on, and you too as well, Teddy, because I think that some people get that impression, and I'm concerned that it will chase them away. So, how do you manage the demands, the apparent demands on time, and raise a family? Obviously, you've done that, Jon. Teddy, you're doing that in this new and more hard-assed environment. How do you deal with it?
Well, I dealt with it by marrying the right person, because I mean, they say you should always be home for dinner. I don't think I was ever home for dinner, right, Teddy? I mean, so I worked really hard, and my wife did a lot of the heavy lifting with the kids. So she worked half-time as a pediatrician, was home every afternoon, and basically dealt with them. And also, my generation was a transitional generation. My parents, we were free range, Vincent and my generation. I mean, you got out of school, you were on your own, right?
Yeah,
and my wife and I were more like that. Our kids were not, we were not helicoptering them for sure. We were not schlepping them much. So it was a bit easier. And what today's parents, they do a lot more for their kids. And I think it's even harder, Rich. And so here's the thing with that curve. It could be linear, but the reason it isn't is because to be really good at science, you have to be obsessed about it, no matter how much you're going to work. And your brain has got, if you're doing creative work, your brain has got to be always thinking about it. And so if that's the case, you're probably going to work hard just because of your obsession, right? I have seen people who have been very successful at 40 hours a week. Usually they're young parents, and their day is highly organized. They do not waste a minute. So, a lot of people spend a lot of time in the lab, but a lot of the time is, I won't say wasted, but it's not directed to achieving reading papers or writing papers or doing experiments or interpreting data. There's other things going on. And I have seen people be successful at 40 hours a week. It is absolutely, definitely possible. But here's the thing. I think if you want a really normal life like most people have, you pick another career. Okay. I just think science is so competitive, and what it takes to have this obsession and deal with it, I think you're going to have to sacrifice some other things in your life. And what I tell young people is you can have, one other thing going on in your life in addition to science. It could be a family. It could be a major hobby or something else, but you can have one other thing, but that's about it, at least until that thing is over. So certainly when your kids grow up and move away, you have more time for lab or whatever you'd like to do, but it does take this obsession, right? And it's not a bad thing. I mean, we went into science because we love it. And that's part of the obsession. It's a joyous obsession. It's like it takes to be a musician. You can't be a musician unless you're completely into it, right? I play music very poorly, and I play with a lot of people who are really good. And so I can see what it takes. And you have a talented person and they play all the time. And it's not work because you love it. And for us, it wasn't really, it's truly, literally a labor of love, right? And I think that then the equation is okay, right? Because someone is actually paying you to do something that you would do for free. And I think that's one of the keys for choosing this as a career is you've got to really love it. And what does that mean? You love doing experiments. You just love designing them and tweaking them and getting the data, doing it better and better. And then of course you love making a discovery that is true. Right? And
Teddy? How about you, Teddy? You're in the thick of it.
Yeah. I mean, I think what my dad first said, your partner, whether it's a wife or a husband, our partner is absolutely key, right? Because they're going to be your teammate in dealing with all this. And then, my wife is amazing at basically, like my dad said, dealing with someone's obsession for something, right? So, and then, you just have to try to tip the scale in your favor, whether you get, you get much more organized when you have a kid. You're a lot more efficient, but we also live 2 blocks from the lab. So
that's great.
I will go home for dinner, at 6. So I can see our son, and then I'll give him his bath, and then if I need to go back to lab, which I frequently do, I'll go back at 7:30, right, or I'll go back at 8:30, and then I can do that. But so it's tough though, because if you want kids, they're going to take a significant amount of your time, and you have to be okay with the fact that potentially you're not going to be able to do as much work as you may think you want to do, whether that's the reality or not. So you just kind of have to equilibrate. And we also, it's we also get a lot of support from our parents, right? So living in Manhattan, our quality of life without that would be, I think, a lot lower. Yeah. So,
what does your wife do?
She's a first grade teacher. She teaches at Heschel. It's on 66th Street.
So my solution was I married a scientist.
Yeah,
right?
that works.
But proximity helps, you know.
Yeah.
I'm 38 miles from home. I was never at home for dinner.
Yeah.
But, you know, she was, so.
Yeah. No, I mean, everyone makes a different deal with their spouse. And for most of us, it works out. And, you know, so another secret to life is that things do not have to be perfect, that they won't be. And everything's about compromising. And you know,
true.
I was in England for 2 years of my life, and English are particularly adept at muddling through things. And I think that works for a lot of life. I mean, nothing's perfect, and you live with some things that aren't quite optimal, and you just get through it. At the end of the day, you know, you got someone like Teddy sitting there and you think, man, my life is worth something. And, it's
not that you want it to be perfect, but you often think it could always be better.
Well,
right?
That's a fallacy. I mean,
it always can be.
Yeah. You know, of course it can be, but, you know, generally I think your happiness that's your responsibility. And we're all born into certain circumstances. And listen, any of us born like we were, the people on this podcast, you know, with families who loved us, who had resources and can educate us. And we are so lucky. And then, you know, also to be alive as a human being at the only time in the history of our species when you could do science.
Right.
Oh my God.
Yeah. It hasn't been that long, right?
No, you know, and, you know, so a couple hundred years if you were a gentleman and you happened to be rich, but for the rest of us schlubs, you know, the children of normal people, we're talking a 50-year window here.
That's right.
Where you're ambitious and you want to go to school and your parents can support you and the government's actually paying money to the universities. This is so we can get lost in the problems in the system. But if you step back, I mean, it is an amazing thing to have the opportunity to be a scientist and make discoveries that will last. If they're true, they will last forever. To put anonymously, it doesn't matter, but just to place this brick in the wall of scientific knowledge is the greatest privilege possible. And I feel fortunate every day of my life. I wake up, it's like, wow, this is fantastic. You know?
To have your micro paper.
Well, to have whatever, you know,
he's also never had to write an R1, so.
That is not true, Teddy. I wrote one.
You wrote one. Okay.
One.
When you were at Whistler.
I wrote one.
Sure,
I got one.
Yeah.
You're right. So, yeah, I have it, and I have a great job.
So you mentioned retiring. Are you going to stop at some point?
No plans. At 70, the lab is going to shrink significantly.
I see.
So, and I think that I want to hang on. We have a great new department chair, Ted Pearson.
I heard that this summer.
Great guy.
Wonderful.
He's a fantastic
So he's
guy.
replacing Bernie Moss.
He's replacing Bernie. Bernie was my chairman for 30 years. Ted is the new guy. It's great working with Bernie, one of perhaps the greatest scientists I've ever known. Bernie.
He's sticking around.
He's sticking around. He's really sharp.
Yeah.
Saw something in our data we missed, and the guy has still completely got it. But Ted has got a lot of new plans, hiring new people, and I am really looking forward to helping recruit these new people and get them going in their career.
Neat.
So that's my plan. So, and my lab will shrink, and I'm losing it. I think we all are as we get older. But creative stuff, I'm still good at. I still can write, and I just love working with the postdocs and the postbacks and the students we have. And being a scientist is basically Peter Pan, right? You stay, in my case, 12 years old, and everyone stays young because they leave as they get older, and it is such a great job.
You're going to get it for one of the best things I can do now that I'm in immunology a little bit is I can get a piece of data. I'll just call them on the phone.
Yeah.
And we'll talk
that's cool.
at any time, 10 o'clock at night, 11:30 at night.
This is really good.
And then we'll talk about that, or we'll also talk about whatever, he's watching Penn State or there's an Arsenal game on. We'll talk about that.
Nice.
But it is really nice.
My students never did that with me. So
and with
great.
Teddy now, the equation has reversed. He gives me ideas to explain our findings.
Nice.
So this is really, it's such a pleasure. My God.
Nice to hear it.
Yeah.
Well, we're going to let you guys go because this has been a lot of fun, but now you have to leave. This has been TWiV 467. You can find it at microbe.tv/TWiV. Send us your questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a number of ways you can help us out. Our guest today from the NIH, Jon Yewdell. Thank you, John.
Great pleasure.
Great to have you back.
Great pleasure.
You should come on more often.
I'm here.
And just call, right?
Yeah.
You come to New York often?
I do, because of Teddy. Yeah.
Let us know.
Yeah.
You're always welcome.
Absolutely.
Teddy Yewdell's at Memorial Sloan Kettering Cancer Center. It's a mouthful.
Yeah.
Thanks, Teddy.
Thanks a lot. This was awesome.
And good luck.
Thank you very much.
All right. We actually started a new podcast called Immune. So maybe someday you'd like to come on.
Oh, great. Yeah, I'll check that out.
We released episode 1.
Oh, fantastic.
Last week.
Good.
So look forward to more immunology.
Yeah.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. He's also on Twitter as Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society of Microbiology for their support of TWiV. And don't forget, for more information about the Department of Microbiology at the Icahn School of Medicine at Mount Sinai, visit bit.ly/micromssm. The introductory music on TWiV is by Ronald Jenkins, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.